Hi and welcome to this video lecture on Sanger's method of sequencing, which is a part of the skill enhancement course on culture collection and preservation techniques. I'm Dr. S. Karthik Sundaram from the Faculty of Microbiology. So in this video lecture, we would be having an idea on what DNA sequencing is all about and the different types of sequencing methods. Apart from that, we would be having an in-depth idea on the methodology and the process of Sanger's method of DNA sequencing. So regarding this DNA sequencing method, what do we know? What do we mean about DNA sequencing? See, the DNA material is composed of just four nucleotides: adenine, guanine, thymine, and cytosine. So, in order to have an idea on the different organisms, identify the organism, classify the organisms, we need to have an idea on the stretch or the pattern in which the DNA is being organized. This mode of identification of an organism is called molecular identification methods. By using DNA, we would be able to identify an organism picture perfectly without trying to go in for a probable method. This gives us a clear method of identifying organism and classify organism based on its sequence strength. So what exactly is the different DNA sequencing methods? We have the Frederick Sanger's methods of dideoxynucleotide sequencing, which we would be having an idea about. This method is also called as the chain termination method. We have the Maxim Gilbert method. This method was actually developed in the year 1977, which is also known as the chemical cleavage method. We have the semi-automated method of DNA sequencing, automated method of DNA sequencing, pyrosequencing, whole genome shotgun sequencing, clone by clone sequencing, and next generation sequencing. Apart from the Frederick Sanger's method and the Maxim Gilbert method, most of the methods were developed in recent times. The time duration that these two methods, both Sanger's method and Maxim Gilbert method, take is quite a large, quite a longer time, and as a result, we have come up to a newer version of sequencing of DNA material. So these newer version of DNA materials has two advantages. One is it can sequence a component of DNA at a faster rate, and the other thing is it can sequence larger stretches of DNA at a very shorter period of time. Say for example, the Sanger's method of dideoxy method. Through this method, we would be able to sequence at a stretch somewhere around 800 nucleotides. But the number of sequencing of the next generation sequencing method is quite higher. So that is the reason why we moved on from the Sanger's method of sequencing method to the next level of sequencing methods. That includes the inclusion of powerful computer aids. So, but before having an idea on what it is, the base of all sequencing method is that of the Sanger's dideoxy method of DNA sequencing, which is also called as the chain termination method. So we need to have an idea on what or how exactly Sanger came up with this alternative, with this procedure of sequencing a DNA stretch. So the timeline here gives an idea about the discovery of DNA duplex and the different years in which we have come up with the different methodologies of DNA sequencing. The discovery of DNA duplex happened in the year 1953, but it was in the year 1977 that we came up with the chemical cleavage method and the chain termination method of Maxim Gilbert and Sanger's. So we have the PCR method or the thermocycler method, which was developed in the year 1985. The first automated DNA sequencing machine was developed in the year 1987. The first capillary sequencer was developed in the year 1996. The Human Genome Project was completed in the year 2003. 2005 is the year in which the next generation sequencer was developed. So much year was in between Frederick Sanger's method, which was developed in the year 1977, and the next generation sequencer, which was developed in the year 2005. So coming to the Sanger's method of dideoxy method. To have an idea of Sanger's method, it was developed in the year 1977. This is also called as the chain termination method. Used as the primary sequencing methodology, it is capable of sequencing only up to 800 base pairs in a cycle. It is time consuming. The basic requirements of Sanger's method is the dideoxynucleotide, PCR reaction mixtures including the polymerase enzyme, DNTPs, and the primers. So what the issue here is that you have a dideoxy structure here. So what is the basic difference between a dideoxynucleotide and the DNTPs? The DNTP has an oxygen in the place of third carbon atom of a deoxyribose sugar. When this oxygen molecule is removed from the entire structure of the deoxyribose sugar, then this becomes a dideoxynucleotide. So how exactly is this dideoxy component going to help us in analyzing the sequence of a DNA? This helps in terminating the growth of a DNA. Say for example, if there is a growing DNA chain, and if you are going to incorporate a dideoxy adenine triphosphate, then the DNA growth will be stopped at that particular point. So this is the component that enabled Sanger to come up with this sequencing method. So to have an idea on what Sanger's method is, we need to have an idea on the PCR process. PCR is nothing but a process through which we amplify a specific region of a DNA. So for a PCR to happen, we need to have the nucleotides. We need to have the polymerase enzyme, which is the Taq polymerase. We need to have the forward and reverse primer and the thermocycler. So during the initial phase of denaturation, the DNA strands breaks, and which happens at the temperature range of 94 to 96 degrees Celsius. And during the annealing phase, you have the primer, forward and reverse primer attaching to the sequences. So during the elongation phase at 72 degrees Celsius, you have the DNTPs and dideoxy NTPs being attached to the growing polypeptide chain through the DNA polymerase, which is the Taq polymerase enzyme. So this is the basis of a polymerase chain reaction. So this is the basic of a basic polymerization reaction, polymerase chain reaction. So how exactly is it going to help us out in analyzing the sequence using the Sanger's method? The Sanger's method has 4 different containers, 4 different measures. The first measure contains the reaction mixture of polymerase chain reaction along with the inclusion of dideoxy thymine triphosphate. The second reaction mixture contains all the reaction mixtures of a PCR component along with adenine triphosphate. The third reaction mixture contains all the PCR mixtures along with the guanine triphosphate. Whereas the fourth reaction mixture contains all the reaction mixtures of a PCR component along with the dideoxy cytosine triphosphate. So these are the 4 components. So what happens inside each one of them? Say for example, we have a DNA stretch here which runs from the 5' to the 3' direction. The DNA which is going to grow will always grow in a particular polarity. It grows from the 3' to the 5' direction. I'm sorry, from the 5' to the 3' direction. So when it grows from the, it needs the 3' hydroxyl group primarily in order to extend its polynucleotide chain. You have the presence of a, so we do have the new nucleotide or the new DNA to which we have the attachment of the primer. The primer attaches here in this particular location at the 3' end. Here it becomes 5' end for the growing DNA chain. So at the 3' end of the primer, you do have the extension of hydroxyl group as far as the third position of carbon atom in a deoxyribose sugar is concerned. So in this growing nucleotide chain, say for example, if we have around 1, 2, 3 thymine residues in this master copy of the DNA. So in the complementary chain, we need to have around 3 adenine nucleotides that needs to be incorporated. Say for example, in the growing nucleotide chain, if instead of a normal adenine nucleotide, if a dideoxynucleotide is added in this from this in the second reaction mixture, what happens is that the DNA length, the DNA will be stopped at this particular point. DNA copy of or the elongation of the DNA will be stopped at this particular point of time. So you have a stretch of DNA of this particular length. Then when the thermocycler runs the second time, see second time, instead of a dideoxyadenine, you have the addition of a normal adenine molecule here. So when it moves to the next nucleotide base pairing, instead of a normal nucleotide, if a dideoxyadenine nucleotide is added here, then you have another DNA stretch of a new length. So now we have added, now we do have two DNA stretches of two stretches of DNA ending with the adenine nucleotides, or two lengths of DNA. So now we have the third thermocycler cycle going on happening here. Now at this stretch, we have another thymine molecule at the end of a fag end of this already existing template DNA. So instead of this, we need to have a dideoxynucleotide or adenine nucleotide being incorporated in this position. So if that is the case, then now we do have three different nucleotide or the three different DNA stretches lengths of DNA that we could develop containing dideoxyadenine as its final element. So same happens within all these reaction mixtures, where in this first reaction mixture, or whatever DNA stretches that ends with thymine, we do have DNA stretches that ends with thymine. So in this guanine triphosphate nucleated mixture, we will have different DNA stretches that ends with the guanine dideoxynucleotide. And in the final cytosine dideoxynucleotide, we would have different stretches of DNA that ends with the cytosine. So what happens? We now do have different stretches of DNA that ends with the specific dideoxynucleotides. When you run this on agarose gel electrophoresis, so depending on the level in which each nucleotides are segregated, we would be able to come up with the sequence of this particular complementary sequence of this DNA sequence. So how exactly is it done? The farthest sequence that moves away from the well forms the first nucleotide. Then we have to calculate accordingly. The next is the cytosine molecule. The third one is the guanine molecule. The fourth one will be the adenine molecule. Fifth one again is another adenine molecule. Sixth one is another guanine molecule. Seventh is the thymine molecule. Eighth is the cytosine. Ninth is the adenine and guanine. Likewise, when we start developing a sequence like this, we get a complementary sequence to the existing template DNA. This is how Sanger developed this method, and through this method we would be able to sequence a particular DNA stretch of around 800 base pairs. So that's all for Sanger's sequencing method of DNA analysis and DNA sequencing. Thank you.